Hello everybody, my name is Bruno Folchi from AgroVentures, an Italian startup company. First of all, many thanks to the IR-4 team for the opportunity they gave us to introduce our YSY Yeast project, and in particular its promising application as biological nematicide. What is YSY Yeast? It's a naturally occurring yeast, ubiquitous globally. We isolated it from the skin of apple and olive fruits in Italy. It works as a strong space and food competitor, growing way faster than pathogens on plant surface, especially where there are exudates, and in particular at root level. It does not produce any toxic metabolite, and it induces systemic resistance and biostimulation. It also gives quite a nice cosmetic appearance by producing some interesting and hexapolysaccharides. We have been developing YSY Yeast as a biological fungicide with plenty of trials in Europe, where we proved its very good activity on a broad range of crops and pathogens, and its flexible use basically at any crop stage from seed to post-harvest. It is a food ingredient, a grass compound safe for humans and the environment. In particular, it is a very strong bee attractant, as therefore application in very sensitive crops like almond, where bees have to be protected. But today we will propose running some trials in the US to test this biological nematode control activity. In fact, while we were testing YSY for soil-borne disease control, we noted a very surprising nematode control activity, which of course added to soil-borne disease control, biostimulation, resistant induction, and great sustainability profile make of these application soil-borne diseases and nematodes together quite an interesting opportunity. Here are some findings about YSY Yeast nematode activity. The data I will show you in the next slides are taken from a publication from a team of 4 Italian universities that are working on testing YSY Yeast performance on soil-borne diseases and nematodes, in particular meloidogene species. You can download their publication from this link. Here I just give you one of their field trials results, so you can see the untreated control was heavily infected by nematodes. YSY at standard rate, 1 kilo hectare, around 1 pound acre, gave around 60% control. Good control, a bit above 50, also at half rate, and overall comparable to the chemical control based on fluopiram, velum prime. Field protocols are quite flexible, up to 6 applications. Water as needed from pre-transplant till the end of the crop cycle. After noticing this good field performance, the R&D team tested at lab level the relative mortality induced by YSY Yeast compared to phosphoazate and abamectin, 2 standards here in Europe. As expected, phosphoazate and abamectin have a fast kill, while YSY was quite slower, basically started acting after 1 week to then catch up with the chemical comparison after 15 days. This was expected because it takes some time for the yeast to colonize the soil media, where it is then quite stable and therefore continues acting over the mid-long term. Interestingly, although kill rate is slow, impact on mobility is quite strong and fast, and comparable to the 2 chemical
products, fosfosylate and abamectin. Basically, we are studying its mode of action, and we know that this interferes with nematodes' mobility, therefore their ability to reproduce and colonize the target crop. As I mentioned, YSY is quite effective to control soil-borne diseases that usually develop in parallel with nematodes. This is an example of some results we had on tomato. 70% control on Pythium and better performance than Trichoderma species. These results are confirmed by a number of other trials and additionally by metagenomic studies indicating that considering soil-borne pathogens reduction where YSY is applied. Just as an example, we know that Rhizoctonia population is reduced by about 6 times after YSY application. We publish this data in the coming months. Registration timeline. We will apply to EPA in a couple of months, and we already applied in Europe where we have emergency registrations. And we are testing the product at field level, in farms level, tomato and strawberry nematodes and soil-borne diseases. Additional information on the product can be downloaded from these 2 web links. Thanks everybody for your attention. All the best.